Good morning, children. So, hope you have all gone through yesterday's videos. If you've got any doubt, please do ask me because the I know that this is it's a little complicated phenomenon or a little complicated pathway when compared to that of the other pathways. So that's why I'm taking it in a slow pace. Okay, so we were dealing with our fatty acid synthase complex, right? So fatty acid biosynthesis, it's happening in a cytosol of cells, right? It's a cytosolic process carried out by multi-enzyme complexes. It's not a single multi-enzyme complex. It's carried out actually two multi-enzyme complexes are involved in the biosynthesis of fatty acids. And for the fatty acids to be synthesized, we should have our acetyl-CoA as the raw material because acetyl-CoA is the starting substance, or it is through the acetyl-CoA condensation that the fatty acids are synthesized, right? So the acetyl-CoA molecules undergoes polymerization to give rise to fatty acids, right? So in the what is happening in the beta oxidation, it is the breaking up of the fatty acid molecule to give 2 carbon units in the form of acetyl-CoA. But it was a mitochondrial process, right? But the exact reverse is happening in the case of synthesis. That is, the liberated acetyl-CoA molecules or the acetyl-CoA molecules which are obtained through various sources will be assembling towards, they are undergoing polymerization to give rise to the C16 palmitic acid. Clear, but it's not a mitochondrial process. It is happening in the cytoplasm. But the problem is that again the acetyl-CoA is residing within the mitochondria. So from the mitochondria, it has to be taken out into the cytoplasm. So it's not that simple because it requires a carrier protein. Because since IMM is not the carrier shuttle system is required, and again acetyl-CoA is not coming in the form of acetyl-CoA as such. It is coming in some other form which is permeable to the IMM. Right, and the permeable form is the citrate molecule or the malate, right? So from the mitochondria, citrate will be formed by the condensation of acetyl-CoA and oxaloacetate to give rise to citrate synthase. The citrate synthase now can easily cross the IMM and come out of the mitochondria and can enter into the cytoplasm where the citrate lyase enzyme, ATP citrate lyase, hence it's an ATP consuming process. So in presence of ATP citrate lyase or consumption of one molecule of ATP, citrate undergo breakdown to give rise to oxaloacetate back and our acetyl-CoA. This acetyl-CoA is now utilized for fatty acid synthesis, while the oxaloacetate has to be taken back into the mitochondria because our TCA cycle has to be replenished, right? So the oxaloacetate again cannot move as such. So oxaloacetate will be converted into malate by malate dehydrogenase. This malate move back into the mitochondria, right? And give rise to oxaloacetate. Right, again the malate, the oxaloacetate can be converted into malate. The malate can be converted into pyruvate by means of malic enzyme. So a part of the malate will be converted directly into pyruvate, or malate itself can cross the IMM and the process can be continued because it is an NAD. Again, fatty acid biosynthesis is a reductive biosynthetic reaction. So a part of the malate will be converted into pyruvate with the help of malic enzyme consuming NADP, so that malate undergoes oxidation while pyruvate, the keto acid will be formed and NADPH will be liberated, and this NADPH is utilized as a fuel, or it is acting as a coenzyme for the fatty acid biosynthesis. So half of the, actually we have, for in order to carry out one cycle of fatty acid synthesis, 2 NADPH molecules is required, right? That is for the condensation of 2 molecules of acetyl-CoA to give rise to 4 carbon molecules of butyric acid. That forms the first step. So each round of condensation reaction, or each round of fatty acid biosynthesis, we require 2 molecules of NADPH, right? So out of the 2 molecules of each cycle, right? Out of the 2 molecules, one of the molecules will be provided by this malic enzyme. The other half is given by the HMP shunt. Okay, so we know that NADPH, the only source of NADPH in the body is the HMP shunt. But in order to spare the HMP shunt by taking or by liberating or by providing all the NADPH required for fatty acid biosynthesis, half of the load is shared by the malate from the that is during this malate shuttle. That's another reason with which the malate shuttle is happening, right? So the malate will be converted into pyruvate with the synthesis of NADPH, so that the NADPH demand can be met by malate also. So half of the NADPH required for example, if for the synthesis of one molecule of C16 palmitic acid, we require how many cycles, right? 2 carbon acetyl CoA is there, so 8 rounds of synthesis de novo pathway has to run, right? So 2 molecules of acetyl CoA enters 8 times, right? 8 round of de novo pathway has to be carried out in order to synthesize one molecule of fatty C16 palmitic acid, right? So out of this 8 pathway, again we require how many number of NADPH? 16 NADPH, because each round consumes 2 molecules of NADPH. So out of the 16 molecules of NADPH which is in demand, 8 of them will be provided by HMP shunt, while the remaining 8 is provided by the conversion of malate to pyruvate, during which the NADPH will be liberated. So that is how the NADPH is provided during fatty acid biosynthesis. So out of the total number of NADPH that is required, half of them will be provided by HMP shunt, while the remaining half is provided by the conversion of malate to pyruvate by malic enzyme. So that is these are the 2 purposes for the running of malate shuttle. The malate shuttle is running, one for the transportation of citrate or oxaloacetate, right, into the cytoplasm for providing acetyl CoA. The other purpose is to provide NADPH for meeting the fatty acid biosynthesis demand of NADPH. So half of the NADPH is given by malate pyruvate conversion, while only the remaining half of NADPH is provided by the HMP shunt. So if suppose X molecules of NADPH is required, half of the X is provided by what? The malate pyruvate conversion, while the remaining half will be given by HMP shunt. Okay, because body is in demand of various other reductive biosynthetic reactions. ഒരുപാട് reductive biosynthetic reactions ഉണ്ട് fatty acid synthesis പോലെ തന്നെ. Clear? അപ്പൊ കൊളസ്ട്രോൾ ബയോസിന്തസിസിലൊക്കെ NADPH, കൊളസ്ട്രോൾ ഫോർമേഷനിലൊക്കെ ഒരുപാട് NADPH മോളിക്യൂൾസിന്റെ കൺസംഷൻ ഉണ്ട്. സോ ഫോർ സ്പെയറിംഗ് ദിസ് NADPH മോളിക്യൂൾസ്, അവർക്ക് കൊളസ്ട്രോൾ ബയോസിന്തസിസിലൊന്നും ഈ മാലേറ്റ് ഷട്ടിൽ അവിടെ റൺ ചെയ്യണില്ല. ക്ലിയർ? അപ്പൊ ആ സമയത്തൊക്കെ HMP ഷണ്ട് വേണം ഫുൾ എന്ത് ചെയ്യാൻ? NADPHനെ കൊടുക്കാൻ. സോ ഫാറ്റി ആസിഡ് സിന്തസിസ് എന്ത് ചെയ്യും? ഒന്ന് അഡ്ജസ്റ്റ് ചെയ്യും. അയാൾക്ക് വേണ്ട പകുതി NADPHനെ അവര് മാലേറ്റ് ഷട്ടിൽ കൂടെ അവര് സിന്തസൈസ് ചെയ്ത് ബാക്കി പകുതി മാത്രമേ എന്ത് ചെയ്യുള്ളൂ? HMP ഷണ്ടിനെ അവര് ബോറോ ചെയ്യുള്ളൂ. Okay, so that is how the NADPH demand is met by the fatty acid biosynthesis. Again, acetyl CoA is formed. This acetyl CoA is taken into the cytosol, and in the cytosol, what is happening? Acetyl CoA undergoes immediate carboxylation reaction by means of acetyl CoA carboxylase, which is a biotin-dependent reaction where the carbon dioxide is coming in the form of bicarbonate ion, and biotin is carrying the carbon dioxide from the bicarbonate towards the acetyl CoA. Hence, this reaction is carried out by a multi-enzyme complex. Acetyl CoA carboxylase is a multi-enzyme complex, that is, it's having more than one number of enzyme activity. One acetyl CoA ne carboxylate ചെയ്യിക്കണം. അപ്പോൾ carboxylate ചെയ്യിക്കണമെങ്കിൽ ആദ്യം carbon dioxide has to come. The carbon dioxide is provided by bicarbonate. So bicarbonate liberates the carbon dioxide. പക്ഷേ carbon dioxide itself cannot go and simply get attached onto the acetyl CoA. കാരണം covalent bond formation ഒക്കെയാണ് അവിടെ നടക്കുന്നത്. അപ്പോൾ biotin picks up the carbon dioxide from bicarbonate ions, so that biotin will be now converted into carboxybiotin. Now this carboxybiotin is releasing the carbon dioxide towards acetyl CoA with the help of biotin carboxylase or carboxybiotin transferase enzyme. So in presence of this particular enzyme, what does biotin do? Biotin liberates the carbon dioxide towards acetyl CoA, so that again there comes a acetyl CoA carboxylase activity. So that acetyl CoA is undergoing carboxylation to give rise to malonyl CoA. So ഒരു എൻസൈമിൽ തന്നെ ഒന്നിൽ കൂടുതൽ എൻസൈം ആക്ടിവിറ്റീസ് ഉണ്ട്. അപ്പൊ acetyl CoA carboxylase എന്നാൽ കാണുമ്പോൾ ഒരു സിംഗിൾ എൻസൈം ആണെങ്കിലും ഒരു സിംഗിൾ പ്രോട്ടീൻ ആണെങ്കിലും ഇറ്റ്സ് ഹാവിംഗ് മൾട്ടിപ്പിൾ ആക്ടിവിറ്റി. ബയോട്ടിനിലേക്ക് ആദ്യം കാർബൺ ഡൈ ഓക്സൈഡിനെ അറ്റാച്ച് ചെയ്യിക്കണം, അതിനുശേഷം കാർബോക്സി ബയോട്ടിൻ ഉണ്ടായി. കാർബോക്സി ബയോട്ടിൻ has to transfer the carbon dioxide from the biotin towards the acetyl CoA. So acetyl CoA should undergo carboxylation. So carboxybiotin transferase and acetyl CoA carboxylase. Yeah. Adim biotin is undergoing carboxylation. Then carboxybiotin is formed. The carboxybiotin undergoes transcarboxylase activity or the transfer of carboxylase. Carbon dioxide has to carry out, and this is carried out by carboxylating acetyl CoA to give rise to മെലനൈൽ കോയ. അപ്പം ബയോട്ടിൻ കാർബോക്സിലേസ് എന്നുള്ള ആക്ടിവിറ്റിയും ഉണ്ട്, ട്രാൻസ് കാർബോക്സിലേസ് എന്നുള്ള ആക്ടിവിറ്റിയും ഉണ്ട്. പിന്നെ പോരാണ്ട് അസറ്റൈൽ കോയക്ക് എന്തുണ്ട്, ഒരു എലോസ്ട്രിക് ബൈൻഡിങ് സൈറ്റ് ഉണ്ട്. സോ ദാറ്റ് ദ എൻസൈം ക്യാൻ അണ്ടർഗോ റെഗുലേഷൻ. The regulatory aspect is through allostric mechanism. So there will be an inhibitor or a stimulator. Appo avarkokka vandirikkan vendittulla cytosine is also there. So it's an allostric enzyme against a multi-enzyme complex having more than one number of enzyme activity, that is biotin carboxylase activity and also trans carboxylase activity. Biotin carboxylase activity carries out carboxylation of biotin, and trans carboxylase carry out the decarboxylation of biotin followed by the attachment of carbon dioxide to acetyl CoA, giving rise to malonyl CoA. Right? So that's one of the multi-enzyme complexes involved in fatty acid synthesis, and this forms a stage 2. That is, first is the transfer of acetyl-CoA from mitochondria to cytosol. Second is the carboxylation of acetyl-CoA to give rise to malonyl-CoA. And the third one is the fatty acid synthase complex. Again, it is yet another multi-enzyme complex. അതിൽ ഒന്നും രണ്ടൊന്നും അല്ല, 7 enzyme activities ഉണ്ട്. ഈ 7 autonomous ആണ്. കാരണം അവർക്ക് ഒറ്റയ്ക്ക് അവർ ബിഹേവ് ചെയ്യും, അല്ലെങ്കിൽ ഒറ്റയ്ക്കാണ് അവർ ഫംഗ്ഷൻ ചെയ്യുക. ഒറ്റയ്ക്കാണെങ്കിലും the product formed during each enzyme activities does not get out of the enzyme. It will be immediately channeled towards the next enzyme, right? So hence it is a multi-enzyme complex, and again all of them are working in a coordinate fashion. This coordinate activity is mediated by carrier protein, which is known as ACP, which is a E7. Clear? From ACP എന്നുള്ള carrier protein ആണ്, അവർ acting as a swing. അതിനകത്തൊരു pantothenate residue ഉണ്ട്. So pantothenate will be acting as a swing, carrying the intermediates from one enzyme to the next enzyme. Clear? So this is fatty acid synthesis enzyme എന്നുള്ള prokaryotes-ലും eukaryotes-ലും ഒക്കെ വളരെ abundant ആയിട്ട് കാണുന്ന, അതായത് it is continued through evolution. ഓരോ evolutionലും എന്തുണ്ട്, evolutionary changes വരുന്നുണ്ടെങ്കിൽ കൂടി it is having no any change, right? അപ്പം it is a conserved gene. Right, the enzyme, the gene coding for fatty acid synthesis conserved throughout the evolution, so that can be found in bacteria and also in higher animals. Right. So what about the structure? We have already discussed yesterday, but I'll be making a recap today also. That is, it's a dimeric enzyme that is made up of 2 monomers. Two set of enzyme units ആണ്. ഒരു 2 E1 to E7 units ഉണ്ടാവും. Clear? So which we call as a structural domains, right? 2 entirely different type of monomeric units. They are joined together, right? And they functions in a single as a single unit. Appo angane varuna samayathu structural division maaritte functional division ennulla form varum. Clear? So it's a polypeptide chain, or it's a polypeptide molecule. It's a single protein molecule having 7 different enzyme activities which are functioning in an autonomous manner. Right? And it's having the 7th enzyme is a carrier protein, which is known as a ACP. So the remaining 6 are acetyl transacylase, malonyl transacylase. In a figure kanikkumbo short forms e undavullu, so you should not get confused. Right? Acetyl transacylase means transfer of the acetyl group. Then malonyl transacylase. So this E2. ഇതിങ്ങനെയല്ല ഓർഡർ, ഞാൻ പറയണമെന്ന് മാത്രം. So malonyl transacylase, what does it do? It transfers a malonyl group. The third one is the 3-ketoacyl synthase, KS. Keto, it is responsible for carrying out condensation reaction. Condense here, 2 carbon units condense here. So it is a ketoacyl synthase enzyme. The next one is, 4th enzyme is ketoacyl reductase enzyme, KR. Right? 3-keto or beta-keto reductase enzyme. Then next one, 5th one is enoyl reductase enzyme. Right? Enoyl reductase. Then comes the thioesterase activity. This enzyme comes only at the final stage of the reaction. Last cycle this enzyme will be coming into action. That is, 8 cycle ഉണ്ടെങ്കിൽ 8th cycle ഈ enzyme will be coming into action. അതുവരെ ഈ 5 enzymes along with ACP will be functioning. Okay. So these are the various enzymes: acetyl transacylase, malonyl transacylase, beta-ketoacyl synthase of 3-ketoacyl synthase, 3-ketoacyl reductase, enoyl reductase, and finally thioesterase. And all of these enzyme activities are coordinated by acyl carrier protein. അപ്പോൾ ഇവിടെ നിന്നുണ്ടാവുന്ന പ്രൊഡക്റ്റിനെ അടുത്തേക്ക് കൊണ്ടുപോകുന്നത് ACP ആണ്. അതുപോലെ മെലനൈലിനെയും അടുത്തേക്ക് കൊണ്ടുപോകുന്നത് ACP ആണ്. ഇവിടുന്ന് ഉണ്ടാവുന്ന പ്രൊഡക്റ്റിനെ ഇപ്പുറത്തേക്ക് ജമ്പ് ചെയ്യിക്കുന്നത് ACP. ഇവിടുന്ന് ഉണ്ടാവുന്നതിന് ഇപ്പുറത്തേക്ക് ജമ്പ് ചെയ്യിക്കുന്നത് ഇനോയിൽ. കണ്ടോ, ഇതൊക്കെ ഒരുമിച്ചാണ് എഴുതിയിരിക്കുന്നത്. റൈറ്റ്? ഇവര് 3 പേരെ ഒന്നിച്ച് എഴുതി, ഇവരെ 2 പേരെ ഒന്നിച്ച് എഴുതി, ഇവരെ ഒറ്റയ്ക്ക് എഴുതി. ക്ലിയർ? വിച്ച് മീൻസ് ദാറ്റ് ഇവര് 3 പേരെ ഒരുമിച്ച് ഫംഗ്ഷൻ ചെയ്യും. റൈറ്റ്? ഇവർ മൂന്ന് പേരും ഒറ്റ ഇതിലുള്ള ഒരു മോണോമറിന്റെ